Okay, I think we can probably get started now. Others will join. So it's my pleasure to introduce our speakers for our research rounds today. We have two speakers today, which is great. We have Dr. Ina Sekirov, who is a medical microbiologist at the BCCDC Public Health Laboratory and holds a clinical assistant professor appointment in our department. Over the course of the COVID-19 pandemic, she's become involved in a variety of SARS-CoV-2 serology-related projects, including serosurveillance initiatives, serological approaches, and their utility for public health and clinical diagnosis continue to hold a special place in her heart. So that will be the overall topic of today's conversation, our talk. And our second speaker will be, I think you're in that order. Is that the way we're going to go? Okay, perfect. Is Dr. Agatha Jassem, who is a clinical microbiologist.
So there's the power.
It's looking pretty good. So yeah, all these things were just like.
It's nice that you're.
There we go. Perfect. Okay, Dr. Jassem is a clinical microbiologist at the program head of the virology lab at the BCCDC Public Health Laboratory. She is a clinical associate professor in her department and oversees provincial diagnostic and surveillance services for sexually transmitted and bloodborne infections, respiratory pathogens, and vaccine-preventable viruses. Dr. Jassem's research interests include testing initiatives aimed at serving hard-to-reach populations and developing surveys in response to emerging viruses. So looking very forward to hear your talk, and I'll turn it over to you.
Thank you very much. And we're going to start screen sharing. So, can you see our slides okay?
That's good.
Yeah, awesome. All right, so as was mentioned, we'll be talking about serosurveillance and how it is really a very vital part of the public health laboratory toolkit. Yeah. Okay, there we go. Before we proceed, we would like to recognize and honor the traditional, ancestral, and unceded territories of the Coast Salish peoples, where we live, work, and provide care, as well as many other ancestral territories upon which British Columbia is located and which our work touches. We don't have any conflict of interest related to this presentation. And in terms of objectives, we would like to go through some descriptions of serosurveillance techniques, define the benefits and limitations of serosurveillance and how it's complementary to other surveillance tools, and then conclude with providing some examples of serosurveys, including for respiratory viruses and emerging pathogens. So in terms of the role of the microbiology lab in disease prevention, we do try to develop diagnostic strategies and surveillance strategies for a number of different conditions and using a number of different methods. So the tools that we have, the techniques that we have, tend to characterize infectivity and characterize transmission. And these are the very basic, information building blocks that we are able to gather. Once we have them, we are then able to link them to such metadata as preventative measures, which underpin, public health policies for these communicable diseases, vaccination, strategies and uptake, treatment outcomes. And together, the lab data plus this metadata constitutes, a form of intelligence gathering. And this intelligence then goes on to inform our surveillance strategy design, which then leads to surveillance strategy implementation. Once implemented, the following data points improve our understanding of these diseases, leading to identifications of disease severity, transmission, prevalence, and outcomes. And altogether, that is what really leads to the betterment of the public health. In terms of surveillance approaches, just a very quick primer on this. So of course, we have the syndromic surveillance, which it was the example of SARS-CoV-2. One of the recent things we've been dealing with was something. Oh my God! Look, there are unusually severe respiratory cases which are creeping up. Something is happening. We need to look into that. Once we know the actual pathogen that's causing a particular syndrome, we can engage in passive surveillance, such as just, really recording the number of cases that show up in a particular hospital, in a particular clinic, or in a particular region. But what we are particularly interested in with respect to surveillance approaches is active surveillance. And that is specifically trying to characterize the strains that are responsible for a particular illness to figure out how much more virus is likely to circulate in one community versus another, what strains are likely to circulate where, and also how this particular virus, actually doesn't have to be a virus, but how does it interact with exposed individuals. And in terms of surveillance methods, what we currently employ in the lab here is genotyping, wastewater, and serosurveillance. And our talk, of course, will focus on serosurveillance, which can be looking at vaccine and or infection-based serological responses, as well as trends in immune protective markers over time. In terms of surveillance samples, there are different approaches that can be taken. We can look at dedicated samples from humans, so human cohorts. The pros for that, of course, is that given that these samples are dedicated collections, it gives us an opportunity to design some very a priori focused cohorts that are able to answer some very particular questions. The cons, on the other hand, are that it does actually take quite a bit of time and effort to recruit these participants, and that requires extra resources. We can use residual clinical samples from human patients. The pros here are that they are quite readily available in clinical labs. And given that we receive large number of these samples, it actually does still allow us to have some stratification based on ages, based on geographical location, and some other variables. The cons are that these are, by definition, convenient samples, and we don't have as much of an opportunity to design, a particular kind of cohort in a particular kind of manner. And then, of course, there are some environmental sample options, with the pros being that given that they're environmental samples, obviously the recruitment issues are much smaller, than when we talk about human people. But the cons are that there is much more variability in sample matrix, and that can affect our ability to design, robust, sensitive, and specific assays. For serosurveillance, the definition of that is testing of blood samples for specific antibodies, and these antibodies can arise due to past infection or vaccination, and it can be in general population or in target populations. So the advantages is that it allows us to capture viral infections that might only have very short periods of actual viral shedding or bacterial shedding for that matter. It can identify prevalence of exposure rather than, like in a more comprehensive way, because, we're able to capture cases that otherwise would have been missed due to, perhaps short period of shedding or just, like not the best window sample collection. It can look at characterizing both vaccine coverage and responses to vaccination, as well as responses to infection. And, over time, and for some organisms, it can be possible to figure out what antibody levels are corresponding to protective immunity and inform protection. So why engage in respiratory serosurveillance? So this here is an image of various pandemics that have afflicted humanity over the last, 130 or 40 or so years. And you will see that 9 out of 13 of them were due to respiratory pathogens. So clearly, we interact with these emerging viruses quite often, and they can create a significant toll on humanity. And WHO, following the SARS-CoV-2 pandemic, has put together a toolkit of how best to be prepared for future emerging pandemics. And as part of the toolkit, they do recommend that labs have access to sensitive and specific assays, including serological diagnostic assays, to be able to readily respond to emerging viruses or other pathogens. I also would like to draw your attention to this paper, which was published in Lancet about a year and a half ago, that brought together a number of people from all over the world who were engaged with serosurveillance initiatives for SARS-CoV-2, and they got together and sat down and kind of tried to synthesize what are the key take-home messages post-pandemic with respect to serosurveillance. And what's key here is that based on their findings, first of all, the number of seroprevalence studies that have happened in response to SARS-CoV-2 throughout the globe were staggering. We had over 4,000 seroprevalence studies. They covered 145 countries, over 34 million people were surveyed. So clearly it indicates that people are very interested in trying to have better, deeper understanding of these serological responses in response to both the virus and the vaccine. And the lessons learned was that really we need to be able to have these assays in place for coordinated responses for future viruses. And keeping and maintaining this infrastructure is going to be key to expand these efforts to other pathogens, as well as be able to respond to newly emerging pathogens quickly. The results of the assays that we will describe to you today, or the results of the studies that we will describe to you today, were often done using this particular kind of test. And so we just kind of wanted to describe the test to you for some background understanding. The test is the mesoscale diagnostic or MSD assay that is essentially like a multiplex serological test. Instead of using individual, wells with a single antigen per well, as is the case with regular ELISA assays, the MSD assay has a number of different antigens for a number of different markers that are affixed to every single well, allowing us to evaluate multiple serological markers at a time per sample. And it's a highly sensitive and specific assay, which has worked out really well for us for a number of different pathogens. So for SARS-CoV-2, just to jig your memory a little bit, the virus itself has a number of proteins which are expressed on its surface. The two proteins I'd like to draw your attention to are the spike protein and the nucleocapsid protein. Both of them are expressed on the cell surface and, virus surface, and are able to generate immune response. With respect to the vaccines that have been deployed in North America and in many parts of the world, all of the RNA and vector-based vaccines only utilize spike as the antigen. So responses to spike can be elicited by both vaccination and infection. The infection, on the other hand, also elicits response to nucleocapsid protein. So if someone has an anti-nucleocapsid antibody response, that is indicative of an infection. So depending on the type of pattern that we see, we either can say that somebody was definitely infected, likely infected if they have any kind of response prior to vaccination campaigns, or likely vaccinated if they only have anti-spike response, but not anti-nucleocapsid response. One caveat to that is that for some reason, the anti-nucleocapsid responses tend to be a bit less robust than anti-spike responses. And every single assay for anti-nucleocapsid that I know of, at least, that has been in use, has demonstrated waning of anti-nucleocapsid response over time. And that is also true for the assay that we have been using. So during COVID-19 pandemic, using serosurveillance has allowed us to have more accurate estimates of infection rates when PCR testing is limited, to have a window into asymptomatic transmission of infection, and to also link our results to administrative data sets on large scale to inform public health responses. The limitations is that depending on the population used for serosurveillance, there could have been more or less bias. Often the results were cross-sectional rather than longitudinal. And again, given that we're looking at the response to an infection rather than the infection itself, there can be time lag to positivity. And with less intense, less severe infections, there will also be less of an intense, less of a strong immune response. So SARS-CoV-2 serosurveillance in Canada was done across the country, and this is an example of sort of an aggregate graph produced by CATF of studies that have covered pretty much every single Canadian province, and looked at the emergence of immune responses to vaccination as well as to infection and how that has evolved over time. In BC specifically, we've had multiple studies as well. One of the first ones looking at serial cross-sectional estimates of vaccine and infection-induced SARS-CoV-2 was performed by Dr. Skavronsky using anonymized age-stratified residual sera from live labs. And so you can clearly see that over the course of the time, this successive waves of infection, you see rising responses to rising infection-induced antibody responses. However, those differed markedly by age group. Some other studies, for example, this one here by Dr. Silverberg et al. Has looked at very specific patient populations, in this case, vaccinated children and young adults. and has been able to demonstrate that South Asian children and young adults have been particularly high risk of infection, and that international travel was associated with higher odds of getting infected. All of these studies together has not just informed our understanding of how the virus spreads, but also has had some significant impacts on, resulting vaccine policy and public health measures. So, for instance, a study by Dr. Skavronsky as well as Dr. Gronau have demonstrated that delayed second vaccine dose was more effective with, inducing stronger protective responses, as well as higher levels of antibody. And these studies have, of course, informed our vaccination strategy in BC, leading to delayed second dose, which has really strengthened our vaccination campaign. More recent study by Dr. Skavronsky linking serological data to administrative data have demonstrated that older adults continue to be underexposed to viral infection and continue to remain at higher risk of hospitalization from infection, which of course needs to be accounted for when sort of designing health utilization strategies. So the pandemic now has moved into an endemic stage. So why should we continue actually surveying, continue serosurveillance of SARS-CoV-2? I'm going to show you some of our more recent data that I'm hoping will convince you that it still has merit and can provide some very useful and actionable information. The studies that I will show you have relied on residual first trimester antenatal screening sample sera that have been collected from every single region in British Columbia and analyzed using the MSD assay. These findings, these results have been really driven by doctors at Valley Marcus, St. Bertari, and Hinsby here on the BC side of things. And the PI for them is Dr. Deborah Money. So this is what the antenatal serosurveillance for SARS-CoV-2 has looked like over the last couple of years. You can see that initially we had, just to orient you first, the blue part of the bars is likely vaccination-induced immunity. The kind of pinkish purple is infection-induced immunity, and the bottom yellow bars are the uninfected, or at least not responsive, not serologically responsive individuals. So you can see that early on, before the emergence of the Omicron, we had high levels of vaccination-induced immunity in the population, but with the emergence of Omicron, infection-induced immunity have also been rising. However, it stabilized for a long time, once Omicron has emerged, up until last respiratory season, when again we started seeing a rise in infection-induced serological responses. Not only did we see higher levels of population become seropositive for anti-N, also even just the levels of anti-spike and anti-N antibodies have been rising over time in the last respiratory season. And when we overlay these responses over the circulating strains, we see something actually quite interesting. So in the 2022-23 respiratory season, the bivalent vaccine that was distributed for BC used B4 and B5 strains as the antigen for the vaccine. And you can see that in that period, like in that respiratory season, the boosters were quite well matched to the circulating strains. However, last respiratory season, 2023, the boosters contain the XBB lineage, the brown lineage, and you can see that this brown lineage has almost disappeared from circulation during the respiratory season. This somewhat kind of like orange-colored lineage was more closely related to the XBB lineage, but wasn't quite it. And even that has disappeared over time. And at the same time, the levels of infection-induced antibody responses have been rising. When we stratify the population by vaccination status, we can see that for the longest time, unvaccinated or red line people here had higher seroprevalence for antinuclear capsid antibodies, infection-induced antibodies, than the vaccinated people. However, last respiratory season, these lines have started to more closely converge. And this is true not just for unvaccinated versus vaccinated stratification, but even when we look at stratification by 2-dose versus 3-dose vaccinated people, the responses have been converging much more closely to unvaccinated people since last respiratory season. Interestingly, though, I'm not going to say that vaccines have been entirely useless or anything of that sort. We can clearly say that the spike seropositivity has been declining. It's sort of the further away you move from your vaccination, last vaccination time point, while the nuclear capsid positivity has been rising with the same sort of periodicity. So clearly, vaccination does, induce seroprotection at some level. And what does this mean with respect to upcoming respiratory season? I mean, I don't have a sort of magic window to look into and say for sure. But when we look at the match of the boosters for this respiratory season, the boosters contain KP2 lineage antigens, and the currently circulating strains are KP3 lineage. The KP2, KP3, I mean, they're not exactly the same, but they're much more closely related to each other than, for example, last year's XBB versus mainly JN1 circulating strains. So I'm kind of hoping that maybe with the closer match, we're going to see, like more protective responses from vaccination relative to the circulating strains. So why should we continue SARS-CoV-2 serosurveillance? It allows us to continue monitoring the newly emerged respiratory pathogen, provides a wealth of future information for future pandemics, and can allow us new insights into vaccine policy and public health policy. And as SARS-CoV-2 transitions into endemic circulation, it can hopefully also inform our knowledge of existing endemic respiratory pathogens and how to interpret serosurveillance initiatives for them. And these are the ones I will very quickly go over now. With respect to other viruses, we've looked at influenza and RSV viruses. For these, you can see that the vaccine targets for influenza are HA and NA. For RSV, I mean, the RSV vaccine is not widely used right now, but the vaccine target is fusion protein F. These are also the same targets that we use for our serosurveillance, so there is overlap between infection and vaccination-induced responses for the detected epitopes. In terms of the methodology, it was actually quite similar to what I just went through for SARS-CoV-2, and the same people have been involved in it. When we look at the findings for influenza serological responses, we can see that as the number of cases rise, like roughly at the same time, the vaccine responses to flu AH3 rise. And that is actually a bit surprising because normally we expect to have a lag between infection circulation and vaccine response rising. So this to me is more indicative of the fact that these responses actually can be elicited by vaccination. For flu H1, it's a similar kind of thing. Again, the cases rise together with, serological responses indicative of like not only infection-induced but also vaccination-induced serological priming. For RSV, on the other hand, this is actually a bit different. So the cases rise first, and the serological responses rise second. And this is actually quite concordant with the scenario where we don't have a widely deployed vaccination for RSV. And I think when we look at these, these case studies separately and together, it can help us interpret what are the potentially protective levels of antibodies and how the viruses interact with the population versus how do the vaccines interact with the population. Similar to what we've seen with SARS-CoV-2, for influenza, people who are unvaccinated tend to have lower levels of antibodies versus people who are vaccinated, which again demonstrates that there is an additive effect of vaccination to seroprotection in addition to exposure to viruses. So for other respiratory virus pathogen serosurveillance, if we engage in that, it will help us understand complexities of immune responses to endemic pathogens, which can then hopefully also help us fine-tune our public health strategies. And the key here is that, because we have a more complicated scenario where the viruses have been circulating and interacting with people for a really long time, we really do need to consider whether or not we need to have more dedicated cohorts versus convenient sampling, and to make sure that we have ongoing database linkages that can help us, understand better the findings that we derive from these studies. And I'll pass it on to Agatha.
Thanks, Ina. So I'm going to shift us away from the respiratory viruses to talk a little bit more about what other applications of serosurveillance are. And so in the lab here, we think about how we can use serosurveillance to understand the prevalence of other emerging pathogens, like we did with SARS-CoV-2 when it was emerging. So there's various examples of these. Some notable ones that we're working on are Mpox and avian influenza, but there are others like arboviruses that we won't talk about today. Other applications of serosurveillance are for vaccine preventable diseases. So you can imagine for something like measles, you may want to understand the immunity gaps a little bit better. So I'll give some short examples and then we'll do some take-home messages. So first, a little bit of Mpox background, because I think people may not be as familiar. But this is, so monkeypox virus, which causes Mpox disease, is a virus that circulates in rodents. It's endemic in parts of Africa, and it is part of the orthopox virus genus. That's important in context of this presentation because it's related to things like the variola virus, which causes smallpox, and the vaccinia virus in that same group. And there are two clades and subclades within those. So in terms of the smallpox vaccine, we have always used a vaccinia-based vaccine. And the current smallpox vaccine that we have stocks for is this modified vaccinia ankara developed vaccine. And this is the vaccine that was used, that has been used in this Mpox current global response. So in Canada, we call it Imvamune, and it's a 2-dose based vaccine. So a little bit about the context, situational awareness, historical and current. So in 2022, I'm sure many of you are aware, there was a big global outbreak of clade 2 monkeypox virus. And this is where, for the first time globally, we really saw extended human-to-human transmission, although it was noted in regions in Africa before this. And so we saw in orange a lot of cases outside of the blue areas, which had endemic Mpox before. And in places like North America, you know, we really saw the spread concentrated among gay, bisexual, and other men who have sex with men. And in some regions, like in Canada, we did have, you know, this vaccinia vaccine administered quite rapidly during this outbreak. So bringing you now quite quickly to the current situation in North America versus Africa, just focusing on those two regions because they are quite different. So in North America, we're still seeing sort of low levels of circulation of this virus. And I put together, you know, some numbers for you to look at there, but still low levels, although in many of our jurisdictions in Canada and BC and Ontario, we did see an uptick of cases to almost daily cases in BC throughout the summer, and it's continuing into the fall. But we still see this largely transmitted through sexual contact among the GBMSN community. This is a little bit, and we did again offer vaccine early on in 2022, and it's still offered to certain populations. In Africa, there are still outbreaks of clade 2B occurring. This is again that major clade that we see here in North America. But there's also more recently been an outbreak of clade 1B. First started in September 2023, so you may be hearing about this in the news. Also looks like evidence of sexual transmission there, and that extended human-to-human transmission. Notably, until the WHO and others, you know, put out public health emergencies just this summer, these regions in Africa really had a hard time getting access to vaccines, highlighting really an inequitable global response to this outbreak, which I think we've seen before. So this is probably why they are continuing to see outbreaks with different variants, whereas maybe some of us aren't, but we're definitely diligently monitoring for clade 1 infection outside of the affected regions, including in BC. So the big question is what's next in North America? So bringing us back to serosurveillance in that context. You know, everyone wants to know, well, what are the risks of outbreaks? What are the risks of outbreaks among MSM? What are the risks of outbreaks among different communities and the general population? And so I just highlight this one CDC modeling and forecasting graph, which basically shows that both for clade 1 or clade 2, where you have vaccines administered, they are highly protective. And so that is a major consideration. But what I think is not captured in these graphs is how much of our population has already been infected. How much of the Mpox spread have we actually seen to understand the immunity from natural infection as well, which will be protective amongst groups. And so there, as with any other infection, we do expect there to be undiagnosed cases. We just don't understand the extent. And with a new emerging pathogen like Mpox, I think this is critical, and this is the role that serosurveys play into understanding the prevalence in this emerging pathogen response. So the role of serosurveys in Mpox, there have been very few studies. We keep looking, there may be one or two more than this, but really few studies doing serosurveys so far, but they're just starting to come out. Some serosurveys were done with pan-orthopox vaccine serology, so it's very hard to have a specific Mpox serology test looking at history of infection because people are also getting vaccinated with Imvamune now or have previously been vaccinated from smallpox, and those viruses are so similar that there's going to be cross-reactivity. So we might call someone false positive for infection unless we do, a diligent job in validating an assay. So previous serosurveys have used sort of pan-orthopox serology technologies, and they've excluded people who were vaccinated for smallpox or with Imvamune. And even during this outbreak in 2022, or just after, places like San Francisco noted an 8% IgG positivity rate in MSM, which is vastly larger than what would have been captured by PCR, which is the diagnostic method. And looking in other populations, even something like 2.5% IgG positivity in homeless. Now with more Mpox-specific serology tests, others are, again, a little bit post this outbreak, showing that a similar number, though, about a 7% IgG positivity in Berlin here, in Rome, in those GBMSM populations. So it brings us to our findings here in BC. So I just want to highlight again someone who was mentioned, Dr. Citlali Marquez, who is leading a lot of our serosurveillance work, but also our funding, which largely came from CIHR, but also some fundings coming now from CERN. And just a lot of the things that Ina spoke to and I'm speaking to are externally funded programs. So first we had to validate an Mpox assay that would let us be confident around detecting exposure to Mpox virus without confusion around it potentially being a false positive result due to vaccinia virus immunization. So again, we're using the same MSD technology that Ina spoke of. We have a multiplex assay there that includes different antigens for the Mpox virus. We used residual blood, so that convenient sampling, from people who previously submitted blood for syphilis testing to the BCCDC Public Health Lab. And we linked that to their vaccination status in the vaccine registry that's available. And we also linked that to their Mpox PCR testing. So we were able to capture groups that were known to be Mpox PCR positive, so should have antibodies post-infection, and the sampling consisted of blood post-infection. We also knew if they were vaccinated or not. For a sort of negative control samples, we had prenatal samples submitted in December 2021 before this virus was circulating in BC, so it should be totally negative, and this group was not offered vaccine either, and should be in the age group that didn't acquire a smallpox vaccine. And we also had sera from people who were vaccinated 2-dose with Imvamune, but were not Mpox infected. So we look at the 5 different antigens, and we're just going quickly through the results and hope there's time for questions. We did find 3 that in particular were pretty sensitive and specific for capturing previous infection with Mpox. E8L in particular, so you can see these antenatal sera, which are negative at the 0, uninfected people, and again, presumed uninfected. We don't actually know if you didn't get a positive PCR test. But lumping around the 0, and then, when blood is collected a number of days after infection, you see this E8L response rising. So that's what we're looking for, versus some other antigens where you can see some false positivity here, like for M1R. So this one worked well. A couple of others worked okay. And, Citlali and others on the team really used this information as a basis to develop cutoffs for this serological assay that would give us a high confidence in our results. Specifically, if you were E8L positive plus 1 or both of these other antigens, we can sort of classify someone based on our validation as being previously infected with Mpox. Now, it's not perfect, so you can see that brings specificity to 95%. So we do think that there is still a little bit of cross-reactivity, but we're using this for population level studies to basically say, well, is the prevalence 5%, 10%, 20, or 50%, or roughly in that range? So we think it's sort of okay. And just want to show you those people who got the 2 doses of Imvamune, where they show up. So specifically, we can look at the 2 doses graph on the right here. So if they were classified as being positive, if they had reactivity to E8L plus or 1 other antigens. So you can see most of these people were blue, green, or yellow, so they were negative. Some of them, again, didn't have history of Mpox infection, but called positive. But again, you can see that only based on our algorithm, 3 out of 22 people gave false positive results. So what I'm going to show you, take it with a grain of salt. There is going to be some false positivity, but really not much. It still gives us in the population level ideas. So then we launched these serosurveys in BC. Here's an epi curve of what's been going on specifically in BC, that major outbreak in 2022. And then, like I mentioned, we do get these sporadic case detections, and we think there is more contributing to this. There's often not any travel to affected regions. And then more recently, we had more activity. So the serosurvey results I'm going to show you come from a period of, here in 2023, after the major outbreak, and some sporadic activity. Then after we sort of had ongoing sporadic activity and a little bit more in the winter. And our future will actually have a serosurvey that's going to be in the fall of this year after this ongoing more activity. So these are samples from males that presented to select STI clinics in Vancouver as our starting point for serosurveys. And we also linked to vaccination data. And so we are breaking these down by age. So because we stopped administering smallpox vaccine in BC in 1972, we are going to look at results for people born before then, as people who may have had smallpox vaccine, which can cross-react potentially with this assay, and then also breaking them down by their other vaccine doses. So the major findings here were that overall in these 2 serosurveys, there was 15% anti-Mpox virus positivity, showing that potentially up to 15% as a whole of these people were seropositive for Mpox infection. And so if you break it down a little bit, you can see it's higher in those born before 1972. And for those born after 1972, which is sort of a little bit more like those studies that I showed you that were already published on serosurveys, it's an 8.5% seropositivity. And if you remember the previous ones, they were 8 or 7%, so quite consistent. We do have that nice negative control of people vaccinated with Imvamune, but we don't really have a good negative control of people who are known vaccinated for smallpox. So that's why this might be higher. There may be more cross-reactivity, and we're just still trying to tease that out. But really, in summary, showing you another application of serosurveillance is that, where we're at here in BC is we do have serology that can identify previously Mpox infected individuals. There may be some cross-reactivity from smallpox vaccination that needs further exploration, but we do note, and even if you just look at specifically unvaccinated individuals, that there's a positivity of 7% in unvaccinated individuals that didn't get Imvamune or likely didn't get smallpox. And an even higher positivity rate actually shown on the last slide, but I didn't elaborate, in Imvamune vaccinated individuals. And that may be because if someone was vaccinated once or twice with Imvamune, they may be at a higher risk of acquiring infection. So maybe less related to cross-reactivity, but more related to risk. But all in all, it says that the prevalence of anti-Mpox virus antibodies really estimates a higher burden of infection than estimated by the acute case detection by PCR alone, and that's really why we do these serosurveys. So next steps for the Mpox work, we will link the serosurvey samples to STI testing and Mpox testing. So many of us wear 2 hats in pathology department. So we want to know, were these people that were seropositive, did they get tested for Mpox, but we missed it? Did they get tested for another STI, but a practitioner didn't ask for Mpox specifically, to understand just the testing landscapes and potential gaps? We want to design our algorithm to look at vaccinia antigens. So there are vaccinia antigens available on the panel too. So we can actually describe vaccine responses as well, not just look for infection-induced immunity. And again, we are doing another serosurvey at this time point. Testing expansion also, first moving to females and then potentially other populations in the future. We're also working with Ontario Public Health Lab so that they can do this type of serosurveys in their province as well. And we're validating on dried blood spot samples. So, we didn't have time to mention, but we've done a lot of work on dried blood samples for many of the pathogens that we're looking at to sort of increase access of testing and sampling. I'll give you one other example, which is on front and center of our minds. I'm certainly in a lot of meetings and was in one earlier today around highly pathogenic avian influenza A. So specifically the H5N1 virus, and specifically clade 2344B, which you don't have to memorize, but this is the one that we're all talking about and that's in the news. So just as a snapshot of a global picture, H5N1 is shown in red. And this is data between October 2022 to September 2023, but it's no different or more now. But we are seeing this virus infect, mostly poultry, but I'll discuss some details. So this is not human cases. This is any instances. So this is avian influenza events. So this is poultry events, but it caused a major outbreak 2 or 3 years ago now, and it continues to spread and return seasonally to these affected areas, including in BC. So where are we at in BC today? It's a busy time for our colleagues in animal health for sure this last couple of weeks, because we did see this virus in the fall of last year and the year before. But we do have a number of infected premises that have really just popped up in the last couple of weeks. So there are current infected premises with this H5N1 clade affecting flocks in BC right now. And then, of course, there have been a number since this outbreak and circulation of this virus has occurred. But there have been no human cases detected in Canada as of late. Where else has this virus been detected? Not only in poultry, as I'm sure you've heard. So detections in cattle, dairy cattle as well. And more recently, a detection in a pig, which actually, had shared facilities with avian species. So let's talk about the human cases, and again, where serosurveys or serosurveillance can be of great benefit. So we can learn from partners who are seeing human infections. So that's in the US. So notably, and I updated these slides, by the way, in the last 2 days, and I think they're up to date, but the numbers are changing very rapidly. So who knows if they're out of date, but in 2024, there have been 39 human cases of this H5 clade. 20 of them have an exposure to infected cattle and 18 exposure to infected poultry, but one case has an unknown exposure and was actually caught by a lab typing surveillance program. And so how could serology help here? And this is just one example, but the US is really expanding their type of serology and other surveillance work they're doing. But for this one case of unknown exposure, the CDC actually looked at their healthcare contacts. This was a hospitalized patient, and they had healthcare contacts, and those healthcare contacts had respiratory symptoms. They were mild, but by the time they used PCR assays to test them, it was too late to detect any virus. And so they actually used serology, and they did find those persons to be seronegative for antibodies specific to the infected virus, which is great news, which means that this person didn't transmit that infection to the healthcare workers. They did find that this person had a household contact who also had respiratory symptoms, and they were actually seropositive for this specific clade and actually the specific infecting virus. And so that suggests that both of them had an infection, even though that one case actually never had a PCR positive results. But luckily, the epi data suggests they had a common exposure and not that there was transmission among each other. But a really good example of how serology can be used. And if you want to use serology for high-path AIV, similar to the other organisms we talked to at more of a population level, that's certainly possible as well. And here's an example. Serosurveillance is not new. We could do a whole talk on historical applications, but here's an example from Vietnam, where again, poultry workers were at highest risk of getting infected with, this is another clade, related clade of H5 at the time that was started to be endemic there, as we're seeing now here. And they actually said, well, we're not getting a lot of PCR positives. Let's do a serosurvey in the poultry workers. And despite not having a lot of PCR positive infections captured, they did have a lot, a significant number, or around the 5, 6, 7% of those persons being seropositive for the infection. So again, it tells us a lot more about the prevalence of infection in an emergent response scenario. So those are the two examples of emerging pathogens. I'll give you a quick example of, where else serosurveillance could be used, and we'd like to see it used more in the BC context, is for immunity to vaccine preventable diseases. So for example, the government of Australia, at least they were doing, I haven't followed up, but every 5 years doing serosurveys using in their population, when a vast range of age groups, looking at what the immunity is there. So assuming that measles there represents actually vaccine-induced immunity, as they have eradicated measles in their jurisdiction much as we have. And they noticed that there was a decline in measles seropositivity among the 5 to 39 age group. So over the courses of the 90s to 2012, and you can see that with the bars going down here. So we do have immune registries, but they're certainly, when talking to our EPI colleagues, they're not complete. So we have information, for example, and this is publicly available too on the BCCDC website, showing that there has been measles vaccine coverage decline by the 7th birthday. So that second dose is supposed to occur by the 7th birthday over the last few years. And so that's quite alarming. But, again, we're relying on vaccine registries for things like coming from school-age children, and we don't understand the immunity gaps necessarily in other populations. So another example where serosurveillance could be used. So really want to just bring it back to some take-home messages, some food for thought, and then open it up for questions. Really, what I hope, what Ina and I hope that you've heard from our, rapid fire, here's a lot of data talk, is really that serosurveillance can be used in a lot of different contexts for a lot of different purposes. But the major impact really comes from linking our data sets. What is just lab information on seropositivity without understanding of vaccination rates, without understanding of dosing rates, as an example, the different demographics of the population, et cetera. So really, when you take the data and you pair it with other data sets and do interdisciplinary work, you can really have some wisdom and hopefully some impact. What I think we have to always be careful of in academic and public health settings is that this doesn't necessarily be taken in the wrong way and actually lead to conclusions that are inaccurate or maybe conspiracy theories by the public. So we do have to watch our communication, but maybe that's a whole other 1-hour talk. One thing that, again, we didn't talk about a lot here, but that's very important to us, is the consideration of the study population. So again, the convenient sample, the residual sera, the focus on specific populations is a good path forward for certain objectives. And I think even in my bio, I said hard to reach populations. And then I thought, oh, no, that's not quite right? It's hard to reach services. And so when we're designing these sero surveys, when we're designing many of the, clinical services and academic work that we do, I think we have to think about, are we missing certain populations that are important based on our study design? So that's certainly something that we're also thinking about for serosurveillance, and that's where the value of dried blood spot collection comes in. And we do try to ensure that we validate this sample type for our assays. And so this is where you can actually take a finger prick of your finger, put it on a specialized filter paper, stick it in the mail, and mail it to our lab, and then we can actually elute that into a buffer and test it on the same platform. So we can do that for some diagnostic testing, we can do that for sero surveys, et cetera. And this way, maybe we can reach other populations that are remote, rural, or don't necessarily want their arm poked for a sero survey. Another thing is, and I think Ina mentioned it a little bit in terms of sort of correlates of protections, but we've been showing you a lot of data that's just antibody levels, and we do recognize the limitations of that. It's a great start, but first of all, we want to characterize the immune responses. There's more to it, and then if we want to really get to understanding what is this sort of cutoff value? Okay, you get maybe a first immunization, a second immunization, maybe it's one exposure. At some point, there's going to be waning antibody, but when are you protected from infection? I'll get to say people are seropositive, but we want to know as a public health organization who's at risk. So that's a big job where we need a lot of smart people to work on. And we also have to consider it's not just antibody level to look at, but the role of functional antibodies, antibody subtypes, avidity determination. And one aspect I tend to ignore, but there's other bright people working on this, is also the role of cellular immunity as markers of protection and immune responses as well. And this is a little trickier because it's easier for us to detect antibodies. The ways that we sample for them, the ways that they can be, again, easily collected on DBS card, not necessarily true for something like when you're looking at T cells. So there's a lot more complexity around there that we need the infrastructure for. So just going to bring it back to, a slide that we often show now at the Public Health Lab is what is our testing toolkit? And Ina touched on this, is really, and it relates to surveillance and also as well is really we have the detection of our pathogens. We have pathogen genomics to characterize them and do that, interdisciplinary work to understand the impact of the genomics, as well as wastewater surveillance and serosurveillance now, as well as this tool that we're trying to build at the PHL for hopefully I've sold you on it and you've seen some good examples. We want to acknowledge so many people. So this is a busy slide and we may have missed people and we, kept adding people. It just really takes a village, a town, entire province and more to put this together. Highlighting some of our, research staff and public health lab staff that work in this serology on the serosurveys with us are noted down here. And also want to mention that we have a lot of BCCDC support and our project managers over there and other supportive staff. We've shown you a lot of the respiratory work, so highlighting some respiratory partners here, as well as the MPOX work, so additional MPOX partners here, and the various organizations who have funded this work, which again has really been externally funded to this date. So with that, we can take questions. Anything else to add?
Nope, all good.
Great, thank you very much, Agatha and for a fantastic talk. I'll open it up. We have a few minutes for questions. If anybody has a question, you can either write in the chat or just speak up. Anyone?
We have overwhelmed you, or I'm not sure.
There's Tyler, maybe he has a question.
Yeah. So I was curious, do you guys know what the timeline is for assays being commercially available to you for new pathogens, like how that works?
Can I start?
Sure, go
Yeah.
ahead.
Well, because so I'll start with just some of the MSD assays, and then I think maybe you could touch more on the other ones as well. But they're pretty quick. But what we have done in the work that we're showing you with MSD is we have actually struck up a pretty good partnership with them, so that we actually talk to them about the type of assays we would like developed, and we work on them together. And even so, they're usually on the ball developing assays such as the SARS-CoV or the coronavirus panel, the MPOX panel. They were in development of those, and we kind of became like a beta testing site for them and had access to those panels before they were commercially available. So although they're pretty fast, we've been lucky to have really quick access to be able to work on these projects quickly.
Yeah, and I would also add that, I think there is also a difference between assays that are commercially available for serosurveillance versus assays that are commercially available for clinical work. Like the serosurveillance ones, they don't need to be Health Canada accredited, and they come out much faster. For COVID, specifically for diagnostic work, because there was so much drive to actually get those, assays into the market, they did become available relatively quickly. But, I don't think we've seen a single Health Canada approved assay for MPOX or anything else that's a bit more, out of the mainstream.
Yeah, that makes sense. 'Cause I remember the early COVID days when there were some studies coming out saying that over half the population's already been exposed to COVID, and it seemed like it was just all false positives. And it was
Well, yeah, there were a lot of assays that you could put I don't know, water on them, and they would give you a positive result. So, yeah.
Yeah, and so there was a lot of that data, and I think in retrospect, with the launching of Serosurvey, some of them did come out quite quickly, and I think you really have to validate them quite well before you launch Serosurvey.
Yeah, oh, that's great. Cool.
Dave?
David has it. Yep.
Yeah, hi. Thanks, guys, both for the presentations. Speaking to your point about things going on Twitter and going sideways, I know, some of the sero prevalence studies obviously got controversial, and I thought one of the, very useful studies that was done was the one that you highlighted, I think, around with Life Labs and that partnership testing residual samples. And that was done across the US for some time. There was coordination with the US CDC, and they had it in almost every state. And when it came out here, people were surprised because nobody else had kind of done that. So there wasn't evidence or data around kids. But I'm hoping that would be something, because that would be one of the first pieces of information be helpful to know whenever we get a new, is understanding what is the true, you know, seroprevalence in the population with reliable assays. I'm just wondering if you think that's something that you'd be able to stand up again, or is that something that other public health labs are planning to do in Canada so they'd be a bit more coordinated? Because I don't know of any others that had tested residual samples like was done here in BC. Certainly not compared to how it was done in the US, from what I understood.
Yeah, so I know that for COVID specifically, there was a lot of residual sample testing done at Canadian Blood Services, for example. But of course, you would miss the pediatric, population with those. I agree with you. I don't think I've seen like the same type of study design as what has been done here with the Danuta's study. I know that for Danuta specifically, she was able to move forward quickly with the use of that live labs collected residual sera because it was a state of emergency, like pandemic emergency, and there were some. It was easier to navigate some of the legal requirements around, like secondary use of these samples. I don't know if it's always as easy for other types of, pathogens that are not, like pandemic pathogens. But I think, actually like working through not just the lab infrastructure, but also some of these administrative considerations, like on provincial level, federal level, that would definitely be helpful. And when you say that it was, this was done in US by CDC, I actually recently was in US for it was called Pacific Public Health Conference. I can't remember the name exactly, but essentially it was for public health related folks in the Pacific side of US, and they also tend to invite BC because we're so close to them. And I was presenting the serosurveillance data there, and what I found actually really surprising was that all of the people who were there, they were specifically state people rather than like federal US people, and none of them had any infrastructure or activity related to serosurveillance in their public health like state labs. It was all done out of CDC, but it didn't translate into state based activity. So I think it's also really key to remember that even though like it's great to like have obviously like federal coordination because it allows us to like tie the information together, but like in their scenario, it didn't seem like that capacity building on the like at the federal level translated into like state level and maintenance of the capacity. So I think it's important to make sure that we actually have local capacity that fits into like wider countrywide information. Yeah.
Thanks.
Great. Any other questions? If not, thank you very much both for a fantastic talk, and